All right. Welcome everyone in the audience who's come to join us here at Neuro Launchpad. My name is Karen Schroeder. I'm a postdoc at Columbia Zuckerman Institute and one of the organizers of this talk series. Thank you for joining us on this historic day. We've come in a bittersweet fashion to the final episode of the first season of Neuro Launchpad talk series, a platform by and for trainees to share and discuss their science. And so far, this season has been incredibly fun and rewarding, and we do plan to continue with future seasons. So quickly, a few announcements about that. So we're going to send out a call for abstracts soon. Stay tuned for that through our website, our listserv, which you can join. There's a link in the chat for that, and also on our Twitter. If you have any suggestions for us going forward into future seasons, we'd love to hear those, so get in touch. And then finally, if you're interested in helping organize also, please get in touch with us. Our contact info is on our website, or you can tweet at us or DM us on Twitter. So today we have two exciting talks on sensory processing in cortex from Chris and Yasir. As you're listening, please do submit any questions you have in the Q&A box. Please don't be shy. We want to have a lot of time for Q&A after the talk, so they can be questions, comments, points of discussion, anything you want to submit. And we would like to hear those. And we're so happy and grateful to have Professor Athena Akrami here to moderate for us today. She's a group leader at the Sainsbury Wellcome Center at UCL, expert in sensory processing, memory formation, learning, and inference, and also someone who's put a lot of effort into creating opportunities for neuroscience trainees, for example, by serving on the executive board of Neuromatch Academy this year. So I think it's very fitting that she's here with us today. Thank you for joining us. I'll give you the stage and let you introduce our first speaker, Chris.
Thank you so much. It's a pleasure and honor to be here. And yeah, it's a bittersweet to moderate the last episode of this series, but hopefully Neuro Launchpad will come back. So the first talk will be by Chris Rodgers. I know Chris from my time at Princeton. So Chris, actually we have a interesting kind of a shared background. He also came to neuroscience from electrical engineering. He did his undergrad in electrical engineering in Purdue, then went to McGill, and then from McGill he went to Berkeley, and he worked with Mike DeWeese on very interesting auditory questions, like a version of cocktail party in rats, and did some interesting experiments and recordings from auditory cortex and prefrontal cortex to show how the animal prepares for kind of to pay attention to different sounds in the environment and do the task switching, and how that preparatory signal manifests in different brain areas. Then after that, he went to Columbia working with Randy Bruno. And I think we are going to hear about that fun, interesting, exciting story that he's been working on for the past couple of years, I think 4 or 5 years. Yeah. So without any further ado, Chris, here we are. Looking forward to hear your talk.
Great. Thank you so much. It's really great to be here, and it's especially great to be introduced by Athena. I've been following her work in rodent decision-making for many years now. I remember back when rodent decision-making was still kind of a backwater at the Cosine conference instead of the Times Square that it is today of so many great future directions. And I also just want to just mention briefly that, I think that, a lot of us are beginning to feel very optimistic now. And I don't just narrowly mean about US politics, but also that globally, I feel that, some things are on the horizon in terms of the pandemic and other challenges. But also I think that it's a good time, even as we're feeling optimistic, to double down on the things that we have learned about ourselves and our community and ways that we can be better. And in particular, I have been very inspired by Athena's moral leadership on a number of difficult issues in our science community and in society at large. And I think that's inspiration for all of us to try harder to do better in the coming year. And I think also, that's kind of the whole ethos of this Neuro Launchpad event, to come together and try to help each other out. So I'm very grateful to the organizers for giving me this opportunity to speak today. So let me go ahead and try to share my screen. All right. Can everybody see my slides now? You can. Yeah, okay, great. So today, the major question that I'd like to talk about is this one. How do we explore the world around us? And how do we recognize the objects that we encounter? So that's something that you and I might use our fingers to do. As you can see MC Escher doing in this self-portrait on the left. And famously, mice and rats use their whiskers in a similar way. So a lot of what we know about perception in the brain, especially at the level of single neurons, comes from experiments in which animals passively experience stimuli. So a picture is flashing on a screen or tones are playing and the animal has no control over what happens next. But that's not a particularly natural paradigm because natural perception is an active process. We choose what motor actions to use to explore the world. And on top of that, natural perception is also goal-directed. So when you look at something, you're usually trying to figure out something specific about it, something that's going to help you achieve your goals. So like maybe you're looking at this slide right now and you're trying to figure out why I'm showing you this painting. Unless you've seen it before. As many of you know, in this classic experiment, Alfred Yarbus tracked the eye movements of human subjects as they examined this painting. And to give them a task, he asked them questions about it. So when he asked the subjects, how old are the people in this painting? Then the subjects looked at faces. So the white lines here show you where the eye movements of the subjects went. And when he asked instead, how old are the people? When he asked instead, how much money does this family have? Then the subjects looked at their clothing and at their possessions. And finally, I really like this one. When he asked the subjects, how long has this family been separated from one another? Then the subjects looked at the lines of gaze that connect the faces in the painting. And so what this tells us is that perception isn't actually just about what's out there in the world. It's about what we choose to explore and what we're trying to accomplish in doing so. So here's how active and goal-directed perception plays out in a tactile system, which is the sense of touch. So this blindfolded woman on the left is identifying objects that are placed in her hand. And the way she does it is to turn them over and over, touch them at multiple points, and in doing so, build up an overall sense of its shape. And similarly, the rat is going to coordinate the motion of his head and his whiskers around objects in order to identify them. And so that's what I want to understand. How do we choose these exploratory motion strategies? And how do we make sense of the resulting sensory input? So this is just the broad outline of this talk, and it also illustrates my thinking about how to approach this project. So first, I'll explain how I defined this problem by designing a shape discrimination task for head-fixed mice. And then next, I'll show how I identified the strategies, or you could say algorithms, that mice use to discriminate shape. And I did this using a new analytical technique that I call behavioral decoding. And finally, I'll show you how that algorithm was implemented by task-specific reconfiguration of responses in the brain. And then I'll finish up with a few future directions that I have planned. So I developed a new shape discrimination task for head-fixed mice. And this upper left image is a little CAD schematic that I put together. So the mouse is head-fixed here in this little house. And I can rotate one of these two orange-colored shapes into his whisker field. And then the mouse reports which one it is by licking one of the two lick pipes that are in front of his face. And on the right is a quick, low-resolution video of the task. So this is all done in the dark. To make sure that he uses his whiskers. What you're seeing is infrared illumination here. I rotate a concave or a convex shape into position. This one is concave. I bring it into his whisker field. The mouse whisks onto the shape in order to identify it, as you see here. And then he learns to lick the left lick pipe for concave shapes and the right lick pipe for convex shapes. Now, because the mouse is head fixed, it permits very precise measurements with high-speed video, which I take at 200 frames per second. These are some example frames from that high-speed video. We're now very zoomed in, so the mouse's head is out of frame down here. And the whiskers that you see here are more or less the same in all mice, and they even have names. So C1 is the long blue one, C2 is tracked in green, and C3 is the short red one here. Now, so far I've told you about a task called shape discrimination. But in the beginning, I mentioned how perception is goal-oriented. And so I wanted to probe that by manipulating the mice's goal. So I also trained a separate group of mice on a different task called shape detection, which is schematized here. Now, during shape detection, the stimuli are exactly the same, but the rule is different. The mouse has to lick right if either shape is presented, and lick left on catch trials when no shape is presented at all. And so having these two tasks allows me to distinguish whether the behavioral and neural effects that I see are just due to the stimuli or whether they have to do with the task that the mouse is trying to perform. So just like the experiment I showed with people exploring a painting with their eyes in the beginning, the mice have the option to explore the same shapes in a different way, depending on what decision that they're trying to make about them. Now, of course, it would be really nice to have the same mice learn both of these tasks and switch between them on command. But I would argue that's not really feasible for a variety of reasons that I'll just skip in the interest of time. But if you're interested in that kind of thing, you can read about my previous work, which Athena mentioned, in auditory task switching here. So this is an example clip of high-speed video during shape discrimination. And this has been slowed down 7 times, so it's all actually happening much faster than this. And I put a lot of work into optimizing the whisker tracking here. Just skipping over the technical details, I can track the whiskers in virtually all of the frames, even as they move rapidly or touch the shape without any jumps or discontinuities in the tracking. And so this is important because the mice tend to make careful, brief contacts with just the fine tips of the whiskers, as you see here. And I didn't want to miss even a single one of those contact events. So I believe that my data set is one of the first high-resolution views of how mice use their whiskers to recognize objects. And as many of you know, the novel object recognition task in mice is commonly used to assess cognitive disorders and therapies. Now, so far, I've only worked with wild-typed, healthy mice. But one potential application of this platform is to determine whether the behavior of a given mouse model is driven by, let's say, a sensory deficit or a motor deficit, or perhaps something more integrative or cognitive. So to understand that a bit better, let's turn to the algorithmic level. So what strategy does the mouse use to convert that pattern of sensory information that you saw in the video into a decision about the shape's identity, concave or convex? I addressed that using a new technique that I call behavioral decoding. I'm schematizing it here with this dashed arrow because it directly relates sensory input, like contacts, into choices without using any neural data. But in doing so, it constrains our hypotheses about how the neurons must perform that same computation, which will be helpful a bit later in the talk. So here's how behavioral decoding works. This is one example trial. I'm showing you the angle of each of 3 whiskers over time. And these colored bars at the top show the times when each whisker was in contact with the shape. So I take all of that data, plus a lot more data that I'm not plotting here about contact force and contact timing, trial history, and so on and so forth. And I provide all of that data together to what's called a stimulus decoder. And that decoder reduces that huge multidimensional data set of features into a single binary prediction. Was the stimulus on that trial concave or convex? And so this basically tells us which features of the sensory data the mouse should be using to identify the shape. I then train a separate choice decoder on the same data, but to predict the mouse's choice. Did the mouse think that it was concave or convex? And this can tell me why the mouse sometimes made mistakes. And for the aficionados, I want to mention that this was based on logistic regression. The innovation here was that I used the error trials to tease apart which features predicted stimulus and which features predicted choice. So just to skip straight to the punchline, it turns out that the way mice discriminate shape is that they compare the number of contacts made across different whiskers. So this analysis that I'm showing you here just shows the behavioral meaning of contacts that are made by each whisker. And specifically, I'm plotting here the weights that the decoder assigns to each whisker making contact, first just for the shape detection task. So during shape detection, the weights are always positive. And that tells us that a contact made by any whisker is evidence that there is something there, which makes sense. But in contrast, here's the weights for the shape discrimination task. Now, positive values means that the decoder thinks the shape is convex, and negative values mean that the decoder thinks the shape is concave. And so we can see that these whiskers now mean totally different things to the animal. So C1 contacts now mean convex, and C3 contacts now mean concave, all else being equal. And this means that the mice are comparing across whiskers, comparing C1 versus C3 in order to discriminate shape. And I just want to mention that all three whiskers can actually touch both of the shapes, but there's a bias in the overall distribution of contact counts, and that's what the decoder is picking up on here. Now, just to summarize the talk so far, behavioral decoding revealed that mice use an elegant strategy to solve this relatively complex task. They compare the number of contacts across whiskers. And I'll put this plot here as a reminder of that. So that brings me to the second part of the talk, which is how could a cross-whisker comparison like this be implemented by networks of neurons in the somatosensory cortex? So before we delve into that, just first a brief introduction to the neural pathways in the whisker system. Sensory contacts are transduced into electrical signals by mechanoreceptors at the base of the whisker. Sensory information flows along this blue arrow, eventually arriving in primary somatosensory cortex, or S1. On the right, if we zoom in on S1, we can see that the columns, the cortical columns, are arranged in a striking topographical correspondence to the whiskers on the face. Early investigators thought that these structures looked like barrels, and so this region is also called barrel cortex. Now, in my study, I found that lesioning barrel cortex severely impaired the mice on shape discrimination, as shown here. So we know that S1 is playing an important role in this task. This slide describes the data set that I collected in barrel cortex of mice performing each task, shape discrimination and shape detection. On the left is a cross-sectional view of the layers of cortex. This green thing is a schematic of the electrode that I use, which allows me to record populations of 30 or so neurons simultaneously across all of the layers. This is a top-down view of barrel cortex. The way that these experiments work is I can target this electrode to any of these columns on any given day. And so I sampled the columns equally, first C1, then C2, then C3, and so on. As some of you know, excitatory and inhibitory neurons can be distinguished in recordings like this using the waveform width, as shown here. I recorded about 1,000 neurons total, and this shows which layer they were recorded in, from the superficial layer 2-3 down to the deep layer 6. And finally, these are the baseline firing rates of each neuron versus its depth in the cortex on the x-axis. So within excitatory neurons, in red, I see the lowest firing rates in the superficial layer 2-3, which is typical of this brain region. This is just an example of what the data look like using this technique. On the left will be a behavioral video like what you've seen before. And on the right will be spike trains from each simultaneously recorded neuron. There's also an audio track that plays the spikes from an example neuron, but I think the audio is not going to work. So I'll just point out visually what I'd like you to focus on. So you can see these rasters flowing on the right side. And you can also see a lot of structure in this neural activity, which I'll point out on the next example trial. These big bursts of activity, like what you're about to see here, that correspond to contacts that are being made on the shape. And then I have another example neuron too, by the way, you can see these videos on bioRxiv if you want to hear the audio track. A second example neuron that responds more to the motor action of whisking rather than to the sensory action of making contact. But in general, even if you could hear the audio, it's very difficult to tell what the neurons are responding to. Because in general, relating the kind of fine scale behavioral data like on the left to this high dimensional neural data on the right is just a big challenge in modern systems neuroscience. So the analytical challenge here, to be more specific, is that at any given moment, multiple whiskers might be touching in complex patterns, plus the mouse might be moving its whiskers in some particular way, plus it may have some idea of what choice it's going to make or a memory of previous trials, and any or all of those things could be driving neural firing. And so to disentangle all of that, I used generalized linear model, or GLM, which is basically multivariate regression. And using this method, I can identify how each of these features affect the firing rate of each single neuron in the data set. And what's unique about my approach is that I'm predicting the number of spikes emitted not just on individual trials or on the average trial, but actually on individual whisk cycles. So I'm really trying to exploit the fine temporal structure of the data in order to understand how all these variables fit together. And I'll just skip these example neurons, showing that the GLM does a good job of predicting neural firing rate. Because I want to just jump straight to the feature that matters most for sensory responses, and that's which whisker made contact. So first, here are spike rasters from a single example neuron again, separately by which whisker was making contact, C1, C2, or C3. And just by eye, you can appreciate how this neuron fires a lot more in response to C1 contacts than it does to C3 contacts. And that turns out to be true not just for this neuron, but for a wide swath of the neurons I recorded, no matter where they were located within the visual cortex. So I'm summarizing that effect in this bar plot, which is the average response to each whisker's contacts averaged over all of the neurons that I recorded. And you can see that the response to C1 at the population level is more than double the response to C3. And that kind of enormous bias was very unexpected in the visual cortex, which is supposed to encode all of the whiskers topographically. And because, of course, I thought maybe I did something wrong, this is where that control shape detection task came in really handy. Because during shape detection, the other task, we can ask how do the neurons respond, and what we found is that neurons respond with no bias whatsoever. And this lack of a bias is exactly what we would have expected given the way that I did my recordings and decades of knowledge about the visual cortex. And so I'll remind you that the shapes themselves and the whisker trimming and everything else was exactly the same between these two groups, discrimination and detection groups. Only the task rule was different. So it's a robust effect, and I have more controls along those lines in the preprint. But what does it mean? Why do the neurons care so much about C1 contacts? And why is it only during the discrimination task? Well, the difference between the two tasks is the rule that the mice apply to those shapes. And if you remember from the first part of the talk, we now actually have a pretty good idea about how that rule should be implemented. So earlier I showed you this plot, which is how the behavioral decoder would have discriminated shapes, just using the number of whisker contacts. And this decoder didn't know anything about neural responses, but it said, in order to identify convex shapes, you should look for C1 contacts and even subtract away C3 contacts. And what I'm seeing in the neural responses is compatible with that. A relative enhancement of the response to C1 compared with C3. So the tuning that I observe at the population level matches the weights that are ideal for detecting convex shapes. So now we can put everything together. The encoder model tells us that neurons have this interesting preference for C1 contacts over C3. And the behavioral decoder shows us that this is useful because essentially C1 contacts are more common on those shapes. And so this leads me to the proposal that sensory cortex is adaptively reweighting sensory input in order to permit an easy readout of object identity. And S1 has learned to emphasize some whiskers over others, that this code is optimized for reading out shape identity. But this is all task specific. So it's the training itself that produces this change in the neural responses. And if you want to read more about that, the preprint is on bioarchive. I just want to have a few slides, if there's time, to talk about future directions, because my long-term vision is to ask, how do we control the motion of our bodies in order to explore and perceive our world? And I think something that is fascinating and unexplored is how we can use head motion to improve our auditory perception. So if you have a cat, you may have noticed that they point their head and their ears towards objects of interest, usually food related. And this is also just a video to show you another example. This is a red fox who is hunting mice hidden under the snow, mice like this one. And the way that the fox finds them is by listening very carefully and hearing their little scratching sounds beneath the snow. And once he knows exactly where that mouse is, he attacks. So I think that landing is really funny. But anyway, there's two things that I want to point out about this behavior. So first is that when the fox carefully listens, he doesn't actually hold perfectly still. He actually cocks his head in a very particular way, as you can see here, left and then right. So he's using head motion to enhance his auditory perception. Now, I will not be bringing these beautiful foxes into the laboratory. Instead, I want to study this in mice who can do something similar. But to study that, I propose a new paradigm, which is to free the mouse from head fixation and ask instead how they freely coordinate the motion of their head with auditory input in order to localize sound. So for instance, I could put the mouse in a circular arena surrounded by speakers, as you see here, and then train it to identify and go over to the speaker that is playing sound. So several groups now have freely moving foraging tasks like this in mice, such as visually guided hunting or olfactory search. But I actually think that auditory is the way to go here, because I would have more stimulus control just by playing different sounds. So for instance, I could make this a little more interesting by adding a little randomness and a little spatial distribution to the sounds that are played, so that the mouse actually has to integrate some spatiotemporal information. And how might this work in the brain? Well, this behavior bridges motor execution and sensory processing. So naturally, I would expect both sensory and motor brain areas to be involved. And in particular, my sort of overall hypothesis is that sensory areas, like let's say the auditory cortex, model the auditory world. So beyond just responding to sound, they would encode things like the goal location and the goal uncertainty in the task on the left, for instance. Whereas motor areas, like let's say motor cortex, model the body, like how the head and the ears can be controlled. And of course, these two systems would have to communicate with each other through inter-area transmissions, which I could monitor and manipulate with optogenetics. Finally, I just want to mention that I think this paradigm could be a good way to study how body motion can compensate for sensory and cognitive disorder. So for instance, people with cochlear implants or kids with ear infections can also use head motions to help them localize sound. So this general model could be a promising framework for understanding a wide variety of computations and disorders in the brain. And before taking questions, if there's time, I would just like to thank my colleagues and collaborators. First, my advisor, Randy Bruno, who was very patient with me and supporting me as this project really took a deep dive from the kinds of sensory physiology that we usually do in the lab to this sort of detailed examination of behavior and population dynamics. Second, I received a lot of guidance from theorist Stefano Fusi and his postdoc, Ramon Nogueira, as I designed and implemented the analyses that I presented to you today. Third, research assistants and future medical doctors, Esther Grieman and Christina Pill, helped me train the mice on this difficult task, which was quite time-consuming and involved. Finally, I would like to thank my funding sources and all of you for your attention. And if there's still time, I'm happy to take any questions now.
Thank you so much, Chris. That is really fascinating. And I'm really happy to hear that you really want to pursue this active sensing in your future lab, hopefully very soon. I don't think there is any question yet. Please post your questions. I mean, while we are waiting for questions, am I allowed to?
I have a question.
Yes, go ahead.
Oh, yes, please. If you do.
Go ahead.
Oh, I have a question. Yeah. So did you find that mice increase the amount of whisking, like the vigor of whisking during the discrimination task versus what is the name of the other one? The identification task?
Detection task.
And yeah, if so, did you find maybe a correlation between that and which mice were better at the task, that kind of thing?
Yes, exactly. I considered many hypotheses, and that ended up being the one that best fit the data. So there were surprisingly few differences between the two tasks. Like if I just showed you a video, it would be very difficult to know whether it was discrimination or detection. And the difference that stood out the most was that, as you put it, vigor. So they would just tend to whisk more times and collect more information for discrimination than they did for detection, which I think makes sense. And I would speculate that actually also, this wasn't significant, but I think that it did correlate with inter-mouse variability and how well they learned the task. So the mice that did better at the task, like it turned out to be actually kind of the most straightforward possible reason. They just moved their whiskers more. They made more contacts, they collected more data, and they did better at the task. Now, why the other mice chose to whisk less is a big mystery to me. Maybe motivation, maybe something else. Yeah, there's a lot of really interesting, I think, variability between mice even performing exactly the same task, similar to results that others have talked about too.
Can I ask a question, which is kind of related? So you talked about the information from whiskers, right? Mm-hmm. Whiskers C1 versus C3, particularly in the discrimination task. Mm-hmm. Which type of information? Which kind of kinematic information is used from each whisker? Did you look at the speed, the deflection, these kind of things?
Yeah, I did. And in fact, that was our hypothesis going in, which turned out to be quite false, was that it would have something to do with some sort of nuanced aspect of the sensorimotor kinematics. So we thought, for instance, the force of the contacts, or especially our favorite hypothesis was the timing, the interwhisker timing, like maybe C1 hits 10 milliseconds before C3 or vice versa. And that was just completely not supported by the data at all. And we did an extensive look at that many different ways. Those variables contain no information or very little information about stimulus or choice, which is quite a distinction from other kinds of tasks that other people have looked at in the whiskers field. And what really stood out over and over was that it's actually just the number of contacts that
just the number.
The number,
so,
yeah.
And the speed of the contact is the same. I'm just trying to understand whether the EFIS data that you observed enhanced firing rate in response to C1 particularly can be explained because just stimulation of C1 was different from stimulation of C3.
Yeah, that's a great question. And so we actually specifically did a control analysis to rule that out. So first of all, I think the most striking thing is that the contacts are not any harder and during one task or the other. And I think you can kind of see in the video too, they're not slamming their whiskers into the objects, which is what people have observed during other kinds of tasks and with other kinds of objects. Instead, they're almost minimally contacting the shape of the object and then almost immediately retracting their whiskers as soon as they can detect that a contact is made. So they're not exploring the full range of follicular forces that they could detect here. They're really doing a very minimal investigate, like a tapping strategy, which is similar to some more ethological reports, especially from Tony Prescott and Robin Grant and people who have looked at this in freely moving animals. That work has not been as well known as some of the work looking at whisker forces in head fixed mice. But I think that my findings kind of support more this idea that it's a tapping motion really, and so the force is just not that informative for this particular sensory parameter. Mm-hmm.
Thank you. So now we have two questions. The first one is by Madine. Do you think the task-specific weighing of C1 is at the level of plasticity or readout? Meaning, would enhanced performance on this task impair performance on another discrimination task where C1 not so useful?
Yeah, there's I think two really good questions embedded in that, and I don't want to take too long, but I think those are both really interesting points, things that I want to look at as in future directions. First of all, I guess I'll just say that the fact that it's C1 versus C3, the reason it's those particular whiskers just has to do with the geometry of this particular task. If it were a different set of shapes, it would be a different set of whiskers that were involved. And I think it would still be a comparison between whiskers, but it wouldn't necessarily be those exact two. That's due to the particulars of this experiment. And I think the other question that's really intriguing is this some plasticity? Is this months and months of training? That's how long it takes. Is that inducing like some thalamocortical rewiring or something like that? Or is it something that's online? And that's something I want to look at in future directions, do some circuit analysis of this. And I'm going to make the exciting, I think, speculation that this is not thalamocortical rewiring, but it's actually online input from other brain regions, and I would suspect the motor cortex, that actually in an online fashion changes these response properties of S1 neurons. Whereas when they're in the home cage or doing anything else, I don't think that I would expect to see the same preference for C1 over C3. But that's something that I'll have to do more experiments to really prove.
That's cool. Okay, the next question, which is also a very good question, it's about, so I just read it. Could you also solve the task with higher C1 and lower C1 bias in the population? It is not clear why C1 has to be higher, given that C1 and C3 are both informative, but just opposite sides, right? In fact, wouldn't an optimal decoding be just using high bias in C1, the ratio of C1 to C3, and low bias in C2?
Yes, absolutely. I think you are correct that would be optimal. And I think what the mice are doing is in some sense suboptimal. In that sense, suboptimal. When I presented this data to Anne Churchland, I told her about this finding, and she immediately said, "Oh, they've turned it from a two-choice task into a yes/no task," which is a concept I wasn't familiar with before. But essentially, it's the difference between kind of doing this equally balanced push-pull, left versus right kind of thing, versus saying, my default is to report it's concave. that's what I'm going to say, unless
exactly.
proven otherwise by collecting enough of the right kind of information to convince me that it's actually the other thing. And for whatever reason, the data that I have, some behavioral data I didn't show you and that neural data that I did show you, suggests mice are using this very particular kind of biased or asymmetric strategy to solve the task for unknown reasons.
That's interesting. Just a quick follow-up on that. Have you tried to introduce some sort of a morphing between the concave and convex? Just like intermediate steps, see how the animal treats those and how that would translate into neural activity.
So these shapes are 3D printed, so I can't modulate their curvature online. I did print some other 3D shapes that were intermediate and present them to the animals, and basically their performance is intermediate on those intermediate shapes, which makes sense. But I think that wasn't really the ideal way to do that kind of experiment. So in the future, I'd like to think about ways to present stimuli that can, exactly as you're proposing, that can be changed online. So in some ways, and that would also be an exciting way to look at, as the animal forms a hypothesis about what the shape probably is, if you can change it at the last second, you could induce very specific kinds of prediction errors. Again, I think this is where auditory will really be a good way to ask these kinds of questions, because it's much easier to just, play a trick on it. Exactly. Yeah.
Yasir has a question.
Oh, yeah.
Can you try to unmute myself?
I can hear you.
Okay. I
think you muted yourself.
Now you muted yourself, yeah.
Okay. You got it? When I try to unmute myself, I go over my name and then my profile. Sorry. I wanted to maybe discuss if you thought about the following. What I got is that they use the information mostly from C1 and C3 whiskers, right? So it looks like they have two reference points in space, and somehow you could yourself try to draw a line, right, between these 2 points. So in a way, have you thought about that instead of shape discrimination, they're doing angle discrimination based on the 2 points where they touch in space C1, C3. In the concave, it will create a line with a certain angle, and in the convex, it will create another line with perhaps a different angle. So it's in a really nice way of reducing the complexity of a task.
Yeah, so the thing that I didn't mention for time reasons is that I present the shapes at one of 3 different possible positions. So they're always presented at fixed locations in space. And I think that although a strategy like what you're describing would still be possible, it's not quite as easy as it might seem, because the shapes can be in different places. And in particular, if I specifically provide the relative angle between whiskers as a parameter of the behavioral decoder, that turns out to actually just not be that empirically, it's not that informative about shape identity, I think, due to the way that I've set it up. And so instead, I think that it's more of a sampling strategy where they're basically just interpreting contacts that are made in a particular region of shape space that's more associated with convex shapes, and comparing that to a number of contacts that are made in a different region of and the advantage of a strategy like this is that it doesn't require making contacts necessarily simultaneously and comparing an exact angle, but rather just taking a kind of a binary output. And so it's, even though what you're describing might be more powerful in some sense, this one might be easier for the mice to actually implement.
So this one is yes, C1, no, C3 versus yes, C3, no, C1. Is that correct? Or did I get it wrong? It's almost like a compare touch versus no touch in the different whiskers.
Yeah, exactly. Compare. Yeah. Now I understand. Yeah, exactly that.
Even easier. It's almost like okay, that's pretty cool.
Yeah.
Very nice.
Just maybe final question before we move to the second talk. Aman is asking, if the mice were able to see the concave convex shapes after being trained, would you expect the mice to whisk in a different manner, perhaps in a way that would predict the shape? Or would you expect them to whisk in the same exploratory way?
If they're exposed to the shapes after being trained?
Yes. I think what he means is some sort of inducing some sort of bias in the way that they are going to explore new objects.
new objects. Yeah, quite possibly. That's an interesting idea. I could present a new object and see how that changes. I think the general flavor of the question is something I'm really interested in. Like, is there something that we get from the way they move their whiskers? Can we kind of get a sense of what they think that the shape is, whatever that shape may be? And there's some hints that they do kind of direct their whiskers to a place where they think that the shape should be based on previous contacts that have been made in the trial. Yeah. That's an interesting question. I'll think more about that.
Okay. So there is another new question. Do we have time for another question?
We're a little, maybe very quickly.
So let's see. It says, Hazal is saying, what do you think would have changed if the shape weren't a set distance away from the mouse? What if instead the convex shape was so close, all the whiskers hit it equally? Oh, yeah. Okay.
Yeah. Yeah, so I think that is actually this is the part that I didn't get into for a time, but actually the way that I've set it up at the closest position, really all three of the whiskers do, or at least can, convex shape. So I think that an interesting point there is that the mice are actually, they're almost choosing which whiskers touch the shape. They don't just kind of throw their whiskers forward and
exactly
as they can, but they actually titrate the way that they move their whiskers, I think actually in a way to kind of bring out this relative difference between C1 and C3. And yeah, so I think that would be my answer to the question, is that
I think that the still animals would adaptively kind of change the whiskers to kind of still manage to pick up different information from whiskers, even if they're all closer and they may touch, but they would just still kind of coordinate the touch such that enhances the differences across whiskers.
Yeah, and I think the way to really answer that question and the previous one would be to kind of introduce some surprise to the animal in the middle of the trial, so we can try to capture, oh, they're trying to target their whiskers in this particular way, but we just pulled the shape back or rotated a little bit. And that is not something I've done yet, but something I'd like to do in the future.
Very cool. Thank you so much, Chris.
Thank you.
Think we are now, we can move to Yasir's talk.
Yeah.
So, okay. So, Yasir Gallero, if I pronounce your name correctly, I'm sorry if I'm pronouncing it incorrectly. He is a PhD student. Chris was finishing his postdoc, and Yasir, I think he is almost at the end of his PhD, right? Yes. So he's finishing his PhD in Zurich, in Brain Research Institute in Switzerland, and on a work that I don't talk about that much because he's going to talk to present that work to us. And before that, he had done a lot of various interesting projects. You have a background in life science in Spain. Right, in University of Malaga. Then you did a master's working with ex vivo patch clamping with Simon Schultz in imperialisms, and then you started your PhD in Zurich. And so I think we're all excited to hear about the work. We actually presented your paper a couple of weeks ago in our lab meeting. So I'm very happy that now I can hear it from you. Very
That's
much
scary.
looking forward. Yes.
Thank you. Thank you so much for the introduction. So actually the time at Imperial was a failed PhD. It's something
no. But it's good that you're talking about that. Failed PhD.
Yeah, so I wanted
you
to
are
bring it
having
up.
a very successful PhD.
Thank you. Yeah, so I not because of Simon. Simon was my co-supervisor. So I basically barely met him. But my other supervisor, it was rough time there, and somehow I managed to survive, start from zero, and
very happy for you. This is a very actually, yeah. I'm glad that you brought it up.
Yeah, I mean, I'm happy to talk more. Sometimes I don't want to mention anything because I almost like can get in legal trouble. But yeah, so yeah, it took some time to recover, and then I started with Fritjof, and then I found like a really nice environment, and everything went really well. I think I was very lucky also to have Ariel Aguilar, the postdoc there. And then yeah, I just defended my thesis on December,
Nice.
and I'm very happy. I think it was yeah, quite a change. And yeah, I just wanted to mention if somebody in the same position. So I'm really happy to talk about it, how I took the decision to finish my PhD, and how did it go with the next round of interviews. Okay, so should it just start?
Yes, thank you.
Right, let me go here, and I think you see my my full screen the right way, right? You see the PowerPoint presentation, correct?
Yes,
Yep, we can see.
Yes, okay, sorry. Yeah, so I'll be talking about a paper that we just recently published in Neuron that is called Sensory and Behavior Components of Neocortical Signal Flow in Discrimination Tasks with Short-Term Memory. And this was been the main work of my PhD. And as with Chris, we are interested in sensorimotor transformation, and in particular, we're interested on how mice can use stimuli from different modalities to guide the same action. In this case, an auditory tone and a texture with certain roughness sensed by the whiskers that will somehow, in both cases, guide to the same action. In this case, a lick. And also a bit on the contrary to Chris, we wanted to focus on the large scale of how the different stimuli from different modality are integrated across primary, secondary, and association cortices. And what I'm showing here is the top view of the mouse brain, showing primary areas across different modalities: visual cortex, somatosensory, auditory, and motor areas. And in particular, we're very interested in understanding the influence of sensory modality in an association area, such as the posterior parietal cortex, or PPC, which, as I draw here, sits in a somehow privileged position between sensation and interaction. And so far, I think mostly the PPC has been considered a multisensory area, some kind of network hub that receives information from sensory areas, V1, S1, auditory, and then sends information to motor areas, but also value-related areas. Somehow, as I mentioned, bridging or selecting the right action in response to a sensory stimulus. But if one pays a bit more attention to its subdivisions, and in particular in mice, then we see that the PPC is mostly constituted by a medial subdivision, or area A, and a lateral subdivision, area RL, that has slightly different connectivity with motor areas, but also importantly with sensory areas. For example, RL receives mostly input from barrel cortex, and V1, and mostly the medial area has connections with the auditory cortex. So we wanted to understand if perhaps mice trained to solve tasks based on different modalities might engage differently these PPC subdivisions. And we also, at the time when I joined, the lab was very interested in understanding short-term memory. So once there is a delay that separates sensation from action in this sensorimotor transformation, that the animal must store behavior-relevant information to bridge the absence of the stimulus until the action is possible. And there have been areas implicated in this, such as the frontal areas like ALM, medial M2, prefrontal areas, which is not seen here in this picture, but also some posterior areas like the PPC, a bit controversial about the involvement, and also some posterior areas that I will also talk about more in the future in a couple of minutes. So with these two questions in mind, understanding the large-scale dynamics during sensation in short-term memory, and how both sensory modality but also behavior strategy, which I will talk in a minute also, how these two components affect and determine large-scale dynamics. We set ourselves to train the same mouse in two tasks, an auditory and tactile task. So these are go, no-go sensory discrimination tasks in which the mice are trained or conditioned to lick in response to the go stimulus only after the delay period where they must hold behavior relevant information. In the case of the no-go stimulus, they are trained to withhold from licking in order to avoid reward. And they also should withhold licking during the delay. So in the auditory task, very simple, they're discriminating between a 4 and an 8 kilohertz tone. And in the tactile task, they're discriminating between a rough and a soft or smooth texture that are presented to the right whisker pad in a similar way as Chris showed. In my case, they're just sandpapers that don't carry any shape, but the dimension to discriminate is roughness. And so the first thing that we noticed, and this was originally published by the Ariel Gilad, which was the poster that also supervised me during my first 2 years in Zurich, is that the animals seem to follow two dramatically different motor strategies during sensation. So active animals, as you see here, during sensation of textures, but also sounds, they move quite drastically, both forelimb and back, even though they are head fixed. And if we extract movement vectors from the back region and forelimb, we see a lot of movement during sensation, and which somehow ends during the delay period and only picks up again during action and reward consumption. Meanwhile, on a passive trial or the passive strategy, the animal is mostly quiet, and perhaps you see a little bit of whisking. So in both cases, you see there he's presented with the same texture, the smooth texture. In both cases, he will lick after a delay period, and in both cases, he will get a reward. So it seems to be a very internally regulated process. We don't control this. Here's how a passive trial looks in terms of movement. There's absence of movement during sensation, but also short-term memory or delay period, mostly movement during sensation. And across this range of activeness from mice that use all the time this passive strategy, we found both of them in both tasks. Some animals that are very passive, meaning 0% of the trials are active, to the other end of the spectrum where animal mostly uses the active strategy. We also found some animals that can interleave and flexibly use both strategies to solve the task. And interestingly, when we train animals first in the auditory task and then retrain the tactile task, some animals change their overall strategy, either decreasing or increasing the strategy. And then to understand large-scale dynamics, underlying this both task epochs, sensation, and short-term memory, we turn to calcium indicators. So basically, there are proteins expressed by neurons that upon calcium entry, which happens every time there's an action potential, they increase their fluorescence. And so we take advantage of transgenic animals that express specifically in layer 2-3 GCAM6 fast neurons, the calcium indicator GCAM6 fast. And what we do is we simply expose the cortex, the skull, which we leave intact, and then we image population dynamics through the skull. What we get is a population signal that looks like this in response to different sensory stimulus. So it has a really nice spatial resolution in the sense that we image all these cortical areas of the left hemisphere with a drawback that we lose cellular resolution. And most of the images that we present there, delta F over F maps, which hot colors represent high activity and cold colors represent low activity. And if we jump right away to how the dynamics looks during sensation in both auditory and tactile tasks, and I show here sense-related areas in the case of auditory areas, but also the PPC areas. And I'm always going to show the same animal trained in both tasks so that we compare really the dynamics within the same brain. So what we see is that in the auditory task, apart from the expected activity in A1, we find a lot of activity in a secondary auditory area, which is called by the Allen Institute auditory dose, or AD. And also very interestingly, we find a lot of activity in the medial part of the PPC, area A, which is the most part of the PPC studied in mice. Most of the studies in mice are in this medial part. Now, conversely, in the tactile task, we see activity now mostly on barocortex, the primary somatosensory cortex, and the secondary somatosensory cortex. But also you see now here on the medial part of the PPC, these areas are mostly relatively quiet, and the shift of activity happens to the lateral part of the PPC with area RL. So with this, I think we conclude that there's a functional segregation in the PPC divisions based on sensory modality, which on average is quite apart, about 1.5 millimeters in space. Since we find these segregated networks of primary, secondary, and association areas, depending on the modality, we ask ourselves if these segregated subnetworks are in a way supported by anatomical connections. And we downloaded data from the Allen Institute, and then we showed that the auditory areas, primary, secondary, and association, or PPC, they mostly connect within each other, with some exceptions. So they connect more within auditory areas than within tactile areas, and vice versa. Tactile areas connect more within themselves than with auditory areas. And this comes to also support and extend previous work that find a nice somatotopy representation of whisker rows in RL, both in anatomy and in electrophysiological recordings. We also did, during the revision, the inhibition of either the medial part or the lateral part of the PPC by using VGAT mice that express general rhodopsin in interneurons, and then we shine light bilaterally in A or RL during the sensory period of both tasks. And so what we find is that if you measure performance here by D prime, being 1.5 the threshold between naive and expert, we see that inhibiting A only affects performance in the auditory task, but not in the tactile task. And conversely, inhibiting RL inhibits the performance only on the tactile task, but not in the auditory task. Now, once that the animal has extracted behavior-relevant information during sensation, and now the stimulus is not there anymore, but somehow they need to store some information to later guide an action, what we see is instead of the sensory modality really influencing cortical dynamics, what we see is that the behavior strategy the animal did, not during delay, but during the past sensory discrimination, is what it will determine the activity during the delay period. I'm showing here an example mouse that during the auditory task was very active and then reduces activeness during the tactile task. And I'm showing you example heat trials, so some that are passive, some that are active. As you see, they might be intermingled in time. And so now if we look at the maps, what we see is that the activity during short-term memory is mostly determined by behavior strategy. And I remind you how the example trial from the active strategy looks. Activity during sensation, movement during sensation, and quiet during delay. So we will see dynamics during this period, the delay. And in the passive, there was no movement in both epochs. And so we see in the auditory and tactile task that the active trials during short-term memory, there's activity in frontal areas, in particular, this medial part of the secondary motor cortex. And in passive trial, there's not so much activity in the front cortices, but mostly in posterior areas that we now term posterolateral association areas, PLA, which are mostly these secondary visual areas, lateral medial, lateral intermediate, posterolateral, and postrhinal. And what we think now, I don't have time to, in a way, support this claim, but we think that frontal activity holds a motor plan, I lick versus I will not lick, in a way, a prospective short-term memory. Meanwhile, activity in posterior cortices during the passive strategy is related to retrospective short-term memory, something related to either the stimulus identity or the value of the stimulus. So in a way, the motor strategy during sensation seems to be determining the location of short-term memory, and in that way, the content of short-term memory, regardless of the sensory modality. Yeah, this figure it's a bit long to describe, but basically we wanted to understand a bit better this change in dominance from sensory modality and to strategy across the task time. And so we compare trials of the same the same strategy but different task, and this is what we call the task index, or how much the task sensory modality or the task itself determines the activity in these large-scale networks. And similarly, we compare trials of the same task but different strategy, or the strategy index, to try to understand, again, how a strategy determines the activity in these large-scale networks. And what we see is that the task index is the highest during sensation, but then it quickly drops, and at the same time that strategy index picks up being highest during delay. So somewhere in between sensation, there is a cross of dominances from sensory modality to the behavioral strategies as the animal shifts from sensation to short-term memory. And we also wondered how does this the route of information from sensory-dependent functional networks, how does this is routed towards frontal cortices in both tasks in the active strategy, or to posterior cortices in both tasks in the passive strategy. And so we think that the parietal cortex might be suited for this, because as a show is required for the task, is very active during sensation, and it projects to both M2 and PLA. We didn't know, however, if there are segregated populations of neurons that project either frontally or posteriorly, or the same neuron can project to both. By injecting retrograde labeling, we saw that most of the neurons either projected frontally or posteriorly. So we think now the PPC might be perhaps responsible or involved for routing the information towards frontal or posterior areas as the task flows from sensation to short-term memory, and it would do so in a strategy-dependent manner. So here is the conclusion slide. We conclude that there's segregation in functional networks during sensation determined by the sensory modality, not only in primary cortices as is expected, but most importantly in the subdivisions of the PPC. And perhaps the PPC itself might be routing the switch from sensation into short-term memory by either routing the information towards frontal areas, holding a prospective motor plan, lick versus no lick, regardless of the sensory modality, or they might be routing the information towards posterior areas in the passive strategy, regardless of the task sensory modality. Right, so here come to the end, and I would like to take the time to thank Fridtjof. I think he really created a good environment in a very competitive lab, which is, I think, something difficult to see. And I'm also very grateful to Ariel, who was there with me the first 2 years. He was a postdoc, now has his own lab in the Hebrew University, and I learned a lot from him. And these are other members of the lab that were involved in one way or the other in the projects. And yeah, I'm very happy to take questions. Thanks for listening, and I hope everything was relatively clear. I'm a bit tired now at this time of the day, also with the baby. So yeah, thank you.
Thank you so much, Yasir. That was really great. And you managed to really make it all these compact information presented in short time. Thank you so much. Do we have any question? No, not yet. Anything not posted yet. But how about you two? Do you have any question before I start? Yes, go, Chris.
This is probably covered in the paper.
You're muted. Yeah. Okay.
This is probably covered in the paper, and I missed it or forgotten. But
That's no problem.
You talked about the decision being held in a motor plan in frontal areas during the active strategy. But could it be something kind of even simpler in that mice, you know, they fidget more when they're going to lick, and so they've actually embodied the memory that they're about to lick in the fidgeting motion? Does that make sense?
So yeah, it does. I think there are some papers about this. So we only analyze the first second of the delay where there's free of movement, at least major movement, and then from that point on, we find the movement and then truncate the trial at that point. So the dynamics are showing also in frontal cortex are movement free, like major movement free. Fidgeting we didn't really analyze, but I don't think what I saw with my eyes is not, they don't really tap. It's true that they start fidgeting and moving towards the end of the delay. That's why to avoid your point, we mostly focus on early delay, the first second, where the animal is actually nicely holding memory with quiet body, and only as the delay progresses, then they start fidgeting and they start moving.
Yeah, I also have a similar comment that just hypothetically, I know you don't have that data, just hypothetically, just try to see how you think about it. Imagine you could record from muscles, for instance, from EMG signals, right? So in these active animals, as Chris suggested, they may just like be more fidgety overall, and they are ready to lick, right? Even if they are suppressing it during the first part of the delay interval, they are so eager to lick. So you would be able to detect that already in the muscle activity, in the EMG signal, in the active group versus the passive group. What do you think about that?
Yeah, I think actually the opposite. The passive mice are worse at holding the memory. They actually have the more passive, the more early leaks. So leaks that they happen too early. And they actually in some of the passive animals we also see a bit of orofacial cortex. So like mouth S1 and perhaps mouth M1. So I actually think that the passive animals are more fidgety. They also perhaps start moving their nose and their tongue. And actually the active animals they are better at holding, both in measured by less early leaks, the more active they are, but also I could analyze this. I haven't done it. Also by feeling they remain quiet for longer. The animals that we train for longer delays, they always were active. So in a way, what we think is actually the opposite, that the movement helps preventing movement. I see this a lot with kids when they are peeing. Right? When they want to pee and you want to hold your pee, you move around, right? So for me, actually, another movement perhaps prevent or help prevent the execution of unwanted movement.
That's interesting. That's an interesting hypothesis. And so, if you just look at the correct trials, not early lick trials, during the sensation, and compare that to the active, do you see any differences in the brain activity of the two groups?
Between I missed it. Between correct and incorrect?
No, only correct trials. Okay, and you look at the active versus passive during the sensation.
Yeah, we do see activity. It's kind of hidden in supplementary figure 4, I think. But yes, so the main thing is that obviously during the active, there's movement and there's widespread activity. There is already activity in M2. There's activity in ALM, and the signal in the sensory cortices is much higher. What we hope to convince is that when you look at sensory-related areas, so barocortex, S2, PPC, even though the activity is higher in the active trials, you still see the same discriminability in passive trials, for example, between hit and correct ejection and and so on. Like if you find like the most quiet trial, the activity, it seems to be confined to primary, secondary, and association. And in the active trial, there's this widespread activity and higher activity in sensory.
So do you think that the passiveness is also somehow correlated with the level of attention and engagement in the task?
Yeah, we always get this comment. Definitely, it's possible.
I'm so sorry.
No, this is the right comment. It's the right comment to ask. I would also do it. I'm just saying that it's a good question. I don't think so.
Do you think there are some ways to tackle that question? Can you?
Yeah,
like,
we could have measured the pupil to measure the
Do we know that like usually like running and movement impacts the pupil, so there might be some kind of a confound just because it's not necessarily attention, but it's some sort of a overall arousal.
What I can talk about is the some evidence against that. For example, there's animals that can switch between the two strategies, even like within seconds, with one trial to the other. Also, passive animals, they can perform very well. So I have animals that they have a really good discrimination, and they're passive. I'm not.
And those animals, even in those animals, the activity during the delay is similar to the active than the passive, or no?
Which animals?
The passive ones that do the discrimination pretty well.
They're similar to the passive. So the posterior activity.
Okay.
Yeah. Actually, look, in the tactile task, there is a nice correlation between activeness and performance, measuring D prime. So the more active, the better you discriminate. Perhaps this has to do with the whisking. So the more whisking, perhaps you have a higher signal-to-noise representation. But in the auditory animals, actually, there's no correlation between activeness and discriminability of sounds. So I'm sure there's an influence of, as you mentioned, this engagement, but I don't think that's all. I think there is some. I think the animals or the motor activity somehow is regulating the type of information that they're extracting. I don't have too much evidence to support this, but this is our current working model.
There is a question in the chat, not in the question box, which is, can you tell more about PLA?
PLA. So PLA is actually a term that we kind of put ourselves. So these are the activity that we find in posterior cortex cannot be really defined to a single area. So we find activity in LM, in the left border of V1, so, I mean the lateral border of V1, LI, also PUR. And in a way, we term them together as posterolateral association areas. What I can tell you about them is that they are classically considered secondary visual areas because they have a complete representation of the retinopathy. They're very nice papers by Burkhalter. But we think they are not only visual, but they're doing something more. For example, there's now work from the Anderman lab in Harvard. And in PUR, which also we consider part of it, he sees a lot of value-related neurons in PUR, and apparently there's a projection in the amygdala. So, I think they're doing something more than just visual-related activity. And now we have even more confidence on this, since in the auditory task, there it's impossible that they are seeing anything relevant to the task. I don't know what else can say about those areas. Yeah. I mean, they connect also to the hippocampus, so there might be also like a kind of the cortical kind of pathway towards the hippocampus and so on.
Have you tried to decode the stimulus from the? I know that with binary stimuli is kind of tricky to do that, but did you try to either increase the number of stimuli, or just like decode the binary stimuli from the from brain activity throughout the different time points and delay interval?
Yeah, so we did a very simple ROC curve and extracted the area into the curve for all areas, for all pixels, between correct trials, so hit and correction. We also did a bit of comparison between trials of the same stimulus but different action, right? So error versus correct, hit versus false alarm, or miss versus correct rejection. We did a bit of that in the paper. And from that, sorry.
No, go ahead. Sorry.
Yeah, from that we may mostly conclude that the activity in PPC seems to be mostly sensory related during early sensation. And in the paper of Ariel that I somehow, I think I mentioned in the presentation in 2018 neuron, he also did something very similar, but for the delay. And this is our reasoning for the sensory related versus motor related. So that the activity in M2 during delay is very similar between hit and false alarm. Meanwhile, the activity in posterior cortex is mostly similar between hit and misses. Yeah, so this is our thinking. I know there are some limitations with the go-no-go tasks. But yeah.
Okay.
That was great. Yeah.
Any question?
Okay.
I think we may have covered it.
Yeah, thank you so much, Yasir. Thank you so much, Chris and Rafaela.
Thank you so much, yeah, to all of you and for the questions, organizers, and you, Athena, really great. Thank you for the introduction.
Thank you so much.
Thank you for being here. Well, we'll
everyone.
be back later in the year with more NeuroLaunchpad.
Yes, thank you.